So good morning. Today we are going to read about typical Mycobacterium. Okay. So in the past class we have already talked about other species of Mycobacterium. So let's start with a broad review of the whole Mycobacterium chapter. Okay. So let's start. So Mycobacterium is broadly classified into two. First, it is like the bacteria which are cultivable and the ones which are not cultivable, non-cultivable. So under cultivable, the first thing which comes is typical tubercle bacilli. So all the Mycobacterium which causes tuberculosis comes under this group, which means Mycobacterium tuberculosis complex. Okay. The second one is which we are going to talk about today is atypical Mycobacteria. So these are found normally in the soil. They are also known as environmental Mycobacterium, and they cause diseases other than TB. Okay. And the last one is your saprophytes. So saprophytic Mycobacteria. So these are non-pathogenic in nature, and they also are found in soil and water. Okay. And under non-cultivable, as you all know, there is only one, which is Mycobacterium leprae, which causes leprosy. So this you have already been taught to you. Also this has been taught to you. So today we are going to talk about atypical Mycobacterium, and saprophytic Mycobacterium is not important because they are non-pathogenic. Okay. So let's first start with the definition. So atypical Mycobacterium can also be known as non-tubercle Mycobacterium or Mycobacteria other than tubercle bacilli, or MOTT. Okay. So there are environmental Mycobacterium which are isolated from environmental sources like water, soil, and also they will be found in birds and animals. Okay. So they are opportunistic pathogens. Whenever opportunity arises, then only they will infect human being. Okay. So opportunity in the sense when an individual is immunocompromised. So individual deficient in cell-mediated immunity is more susceptible to get this disease, and there is no human-to-human transmission in atypical Mycobacterium. Okay. Then other than that, there is saprophytic Mycobacterium. As I already told, they are non-pathogenic and they are found in soil and water. There are two examples, which are M. fili, which is found in grass, and smegmatis, which is found in the smegma. Now smegma is your dead cells and all this fluid accumulation in the tip of the penis or the folds of the vagina. So they are normal contaminant found in the urine. Okay. So now atypical Mycobacterium, they were classified by Runyon in the year 1959 based on the pigment production and the rate of growth. So based on pigment production, they were divided into three, which is photochromogens, scotochromogens, and non-photochromogens, and based on the rate of growth, rapid growers. Okay. So what are photochromogens? So photochromogens are the are the Mycobacterium which produces pigment only in the presence of light. Okay. Examples include Merinum, Asiaticum. We are going to talk about each of these in the following slides. Okay. Now then there is scotochromogens which produce pigment both in dark and light. So in dark you will see a pigment. When you put light in it, the pigment is going to intensify itself. Okay. Then there is non-photochromogens which do not produce any pigment, and then there is rapid growers which grow within a week. Okay. So photochromogens, the Mycobacterium which produces pigment only in the presence of light. Okay. So first example is Mycobacterium marinum. Okay. So these Mycobacterium, they grow at a temperature of 33 degrees centigrade, and they will grow poorly at 37 degrees centigrade, or they may not grow also in that temperature. Okay, they are acquired only through water sources, mainly fish tanks and swimming pool, and they enter into our body through minor trauma. So if there is a minor cut in the body, a person gets inside the swimming pool, and if the mycobacterium is there, he is going to get infected. Okay, so infections lead to the formation of lesions. Okay, so lesions include papules and ulcers, which are known as swimming pool granuloma or fish tank granuloma. Okay, then there is tendonitis, and then tender nodules are also seen, which spread in a sporotrichoid pattern. Now, sporotrichoid pattern means there is a dimorphic fungi known as Sporothrix schenkii. So when infection through this fungus occurs, there are nodules which spread in a particular pattern, which is known as sporotrichoid pattern. Okay, so similar to that, M. marinum also produces nodules in the same pattern. Okay, so this is a picture of the pattern. Okay, this like this, there are nodules here, and there is sporotrichoid pattern here. Okay, and then another one is Asiaticum, which is rarely associated with pulmonary disease and bursitis. Okay, now as you can see in the dark, there is no pigmentation in case of photochromogens, and there is in light there is yellow color pigmentation. Now this is the growth in LJ media. The colonies are again pigmented yellow. Okay, this is Mycobacterium marinum. Okay, then we come to Mycobacterium simiae. Now simian flu, simiae means monkeys. Okay, so they are isolated from monkey. They give positive niacin test. Now the other Mycobacterium which gives positive niacin test is Mycobacterium tuberculosis. So it is often confused. When we do niacin test, two organisms is going to come positive within Mycobacterium tuberculosis and simiae. Okay, so the other tests are required to differentiate it from Mycobacterium tuberculosis. Okay, so they are principally isolated from pulmonary lesions. Then there is Kansasi, which causes chronic pulmonary disease which resembles tuberculosis. Risk factor is the old person which have pre-existing lung disease and impaired immune response. Okay, they are again isolated from soil, water, and milk. Also milk here. Remember, Kansasi is isolated from milk also. Okay, so they grow at 37 degrees centigrade on LJ media and produce yellow to orange pigment. Okay, then there is Genavens, which grows slowly and is associated with patients which have HIV, who have advanced HIV. So again, immunocompromised. Okay, now we come to scotochromogens. Now scotochromogens are, when you incubate them in dark, they are going to produce colors like yellow, orange, and red. When you take it out and put it in light, then the color is going to intensify. Okay, so examples include scrofulaceum, which causes scrofula, which is a lymph node disease. Okay, basically it happens in children, and it is found as a common contaminant in the clinical samples. Okay, now they are also associated with pulmonary diseases. Okay, then there is Suzel-Guy, which behaves as a photochromogen at 37 degrees centigrade, at scotochromogen at 37 degrees centigrade, and photochromogen at 25 degrees centigrade. So at 25 degrees centigrade, they are going to produce light color only in the presence of light, and at 37 degrees centigrade, they will produce color both in dark and light. Okay, now they also are associated with pulmonary disease and bursitis. Then there is Mycobacterium cellatum, which is again associated with pulmonary infections. Okay, so this is the image. So when you incubate when you inoculate the organism in LJ medium and incubate it in dark, then also they are going to produce this yellow type of color. So this organism is Mycobacterium scrofulaceum. Okay, and then scrofula, this is how it looks. There will be inflammation of the lymph nodes. Okay, and it majorly happens in kids, in children. Then we come to non-photochromogens, which do not produce any pigment. So examples here include MAC. So MAC is Mycobacterium avium intracellulare complex. Okay, So it contains of two bacteria, Mycobacterium avium, which is isolated from birds, and Mycobacterium intracellulare. So it again is a opportunistic pathogen, is going to cause disease in immunocompromised patient who have HIV or who have low low CD4 T cell count, okay, lower than 50 per microliter. So it is again associated with lymphadenitis, which is a lymph infection. Then there is respiratory infection, and then disseminated infection, which can cause osteomyelitis also. Then we come to Xenopy. Xenopy is isolated from hospital water supply and is associated with mainly nosocomial infections. So it is a normal commensal and it causes pulmonary disease in again HIV infected immunocompromised patients. So it comes under non-photochromogens, but sometimes Xenopy may form yellow color colonies and similar to, and so they can be also put under scotochromogens. Okay. Then there is ulcerans. So in your non-photochromogens, ulcerans is very important. Okay. So they are waterborne skin pathogens. They are found normally in tropics of Africa, Central South America, and your Southeast Asia. So the main thing which this causes is Buruli ulcer. So Buruli is a place in Uganda. So that's why the name. So it forms a painless ulcer, and which later can turn in necrotic. Okay. So when it turns necrotic, it can disseminate to other parts and may cause osteomyelitis and lymph deformities. Okay. Now, what is the exotoxin here is myolactone toxin, okay, which helps in the pathogenesis. They have a narrow temperature range of 31 to 34 degrees centigrade. They grow slowly, and the colonies will appear for after 4 to 8 weeks. Okay. So this is your Mycobacterium ulcerans. As you can see, there is no pigment here in the colonies, and this is your ulcer, Buruli ulcer, which is a cutaneous infection. They will be painless and they can disseminate. Okay. Okay. Now we come to other examples are M. malmoense, which again causes pulmonary disease and rarely lymphadenitis. And then there is paratuberculosis, which is also known as John's bacillus, which causes disease mainly in cattle and is associated with pathogenesis of Crohn's disease. Okay. Other non-photochromogens include Chimoidae, Terrae, and Gastri. Okay. Now the last one is rapid growers. So rapid growers, as we are already told, it will grow within a week. All the others, they will take too much time to grow, minimum of 4 weeks to grow. But this, they will grow within 1 week. So examples include Fortunum and Chilone, which causes post-trauma injection abscess and catheter-related infections. Then there is abscessus, which again causes pulmonary infection. Now to identify rapid growers, there is one specific test known as Aryl sulfatase test. It will only be positive for all the rapid growers and will be negative for all typical Mycobacterium, atypical Mycobacterium, and non-cultivable Mycobacterium. So this is your specific diagnosis for this test is done for the specific diagnosis of the rapid growers. Okay. Then the principle of the test is that there is a chemical known as tripotassium phenolphthalein disulfide or sulfate. If Aryl sulfatase enzyme is present in the Mycobacterium, then it will break this and form free phenolphthalein, which will change the color of the medium from colorless to pink, okay, which will show positive test. So Mycobacterium fortunatum and Chilone will be positive, while avium, which was your non-photochromogen, will be negative. This is a rapid grower. Okay. So other test to differentiate them is your growth in McConkey agar. Now Fortunum and Chilone can be differentiated through this. Now if you grow the organism if you inoculate these two organisms in McConkey agar, which will have 5% NaCl, then Fortunium is going to grow there. While growth in McConkey agar. So if you inoculate both Fortunium and Chelone into McConkey agar in the presence of 5% NaCl, then Fortunium is going to grow while Chelone is not going to grow. So this test is done to differentiate between the two. Okay. Now we come to the clinical manifestations. Okay, so this is the important chart. Now all this which I've summed up all the things in one chart, so it will be easier to remember for you guys. Okay, so first pulmonary disease, which will so what are the infections which atypical Mycobacterium causes? So first is pulmonary infections, then lymph node infections, then cutaneous infections and disseminated infections, and these are the organisms associated with it. Okay, so first in pulmonary we see MAC, then Kansasi, then Xenopi, then Malmonese and Sulgani and Abscicus. Now MAC is common in all the infections. Okay. Then in lymph node infections, as I told, Scrofula was a lymph node infection caused by Scrofulatium, and again Malmose also causes lymph node infection. Cutaneous infections, we talked about your fish tank granuloma or swimming pool granuloma, which was caused by Mycobacterium marinum. Then we talked about Buruli ulcer, which was caused by ulcerans. Then abscess, so abscicus, which is also a cutaneous infection. Then Fortunium and Chelone, which were the rapid growers, they cause injection abscesses, and then again MAC. Then disseminated infections like osteomyelitis and all, they were caused by MAC and Kansas. Okay. Now we come to lab diagnosis. So according to these infections, if there is pulmonary infection, then we are going to get sputum sample. If there is lymph node infection, then we are going to get lymph node aspirates. If there is cutaneous infections, we are going to have pus or exudate, and if there is disseminated infections, then we can get the biopsy. If there is skin lesions, if there is skin lesions, we can get biopsy, and in disseminated infection, whichever side it is being disseminated to, that particular sample we might get in our lab as a specimen. So sputum, lymph node aspirate, pus and biopsy are the usual specimens. So first thing we do is microscopy through Zn staining. It shows your red acid fast bacilli. Now these bacilli will be similar looking as of Mycobacterium tuberculosis, so they need to be differentiated by performing other biochemical tests. Okay, so this is how they look like. Now culture in LJ media, so all these NTM or non-tubercle Mycobacterium or atypical Mycobacterium grows well in LJ medium, however few will go sparsely also. Now pigment production, LJ media are inoculated in light and dark separately, so that you can differentiate between photochromogens and scotochromogens. Photochromogens will only grow in produce pigment in light, while scotochromogens would produce pigment both in dark and light. Okay. Then catalase test. Now catalase test is very important for this. So atypical Mycobacterium, generally all of them will be catalase positive. So we do two types of test here: semi-quantitative catalase test and heat stable catalase test. Now in semi-quantitative, what we do, we will take a test tube, we will inoculate the NTM organism colonies here, and then in this we are going to add H2O2. Okay, so by that we are going to see there will be obviously there will be effervescence. So here in semi-quantitative catalase test, we measure how much the effervescence is. So if the bubble is rising here, we are going to measure this, how much what is the height of what is the column of bubbles, we will measure the column of the bubble. Okay, so if it exceeds 45 millimeter, then it is considered as a NTM colony. If it is lower than that, then it is not an NTM colony, not a atypical mycobacterium. And other test is heat stable catalase test, where we heat the culture at 68 degrees centigrade for 20 minutes. And then if we take this culture and perform catalase test, again it is going to show effervescence. Okay, others won't show effervescence if the culture is heated to this much extent. Okay, so this is how it looks like. So catalase, this is a like control, and then when we put organism here, if the effervescence is only this much, which is less than 45 millimeter, then it is not a NTM or atypical mycobacterium. If it exceeds, so this will be your 45 millimeter. So these two are NTM colonies. Okay. Now, all these typical tubercle bacilli and atypical mycobacterium needs to be classified based on certain biochemicals. Okay, so the first biochemical is your niacin test. Now in niacin test, we look for nicotinic acid, production of nicotinic acid. So it is always positive in the case of Mycobacterium tuberculosis. And one more organism I just told it is positive for, which was Mycobacterium seminae. Okay, seminae. So as you can see, 3 tests are performed: niacin test, nitrate test, and pyranamidase test. Okay, so it will be positive for Mycobacterium tuberculosis and Mycobacterium bovis, which comes under Mycobacterium tuberculosis complex, will be negative for atypical Mycobacterium niacin test, and nitrate and pyramidase test will be variable for different species of non-tubercle Mycobacterium. Okay, then we come to tween 80 hydrolysis test. Now in this test, what happens? There's a we check for the presence of lipase enzyme. So if tween 80 is there, and if lipase enzyme is present, then it will break tween 80 into oleic acid. So Mycobacterium tuberculosis will be variable, Mycobacterium bovis will be negative, and NTM will be variable in this case. Then we come to optimum temperature. Mycobacterium tuberculosis complex will grow at 37 degrees centigrade, while atypical Mycobacterium, under atypical Mycobacterium, ulcerans would grow at 32 degrees centigrade, marinum at 30 degrees centigrade, chilone at 25, and xenope at 42 degrees centigrade. So optimum temperatures is variable in case of atypical Mycobacterium. Okay. Then we come to catalase test. As we already talked about, there will be 2 catalase tests in it. Semi-quantitative catalase test, where Mycobacterium tuberculosis complex, the bubbles won't rise till 45 millimeter, while in atypical Mycobacterium, bubbles are going to rise more than 45 millimeter, which will show positive test. Then heat stable catalase will be negative here and will be positive for most of the species here. Then there is aryl sulfatase test, which will be negative in case of Mycobacterium tuberculosis and will be positive in case of only rapid growers under atypical Mycobacterium. Then we come to growth on Mekongki agar with the addition of 5% NaCl. So again, your typical Mycobacterium will have no growth here, while only Fortunum and Abscus is going to grow. Okay, now last we come to treatment. Now, similar to Mycobacterium tuberculosis, non-tubercle bacillus also needs multi-drug treatment because it is associated with emergence of drug resistance and relapse. So in case of Mycobacterium avum, intracellular complex, and marinum infections, we give multi-drug therapy, which includes macrolides like clarithromycin and azithromycin and rifamycin. And in case of cansasis, we give isoniazid, rifampicin, and ethambutol for prolonged duration. Okay, so thank you.